Today's topic is a DNA structure and replication. So DNA structure I'm going to go over pretty quickly because we should be confident with this by now. But looking at our picture up here, we can see DNA shape is a double helix or like a twisted ladder. I can see each side is made up of repeating sugar and phosphate. So this is our sugar phosphate backbone made up of strong phosphodiester bonds. In the middle, we can see our nitrogen bases linked together following Chargaff's rule. Cytosine pairing with guanine, adenine pairing with thymine. We can see our weak hydrogen bonds in the middle. So as a reminder, DNA is a nucleotide falling under the macromolecule of nucleic acids. With this, we can see a great shout out to our amoeba sisters making this GIF with our sugar, phosphate, and nitrogen base. If you have a hard time with DNA replication after this video, I'm 100% looking at the Amoeba Sisters DNA replication. They do a great job of showing this tricky process. And I actually took some screenshots of their pictures and we're going to explain it through their work because they do an amazing job at this topic. So our DNA strands we know are anti-parallel. So in class we related anti-parallel as driving opposite directions down a road. So you're running next to each other, but you're going the opposite direction. So our DNA strands are the same. They're anti-parallel. Strand 1 will be facing one direction. Strand 2 would be facing the other direction. So with this, it's important to remember how we count our carbons and how we identify each strand. So I always start at the top here where the oxygen is. I think of the oxygen as 0. And you always count clockwise. So 0, 1, 2, 3, 4, 5. So reading this top to bottom, this would be our 5' to 3' strand. We can see that oxygen is facing the top. So we call the 5' to 3' our leading strand. And then our 3' to 5' will be our lagging strand. So these are going to be coded differently when it comes to DNA replication. So with DNA replication, think of it making exact copies. So if I were to put a worksheet on the copy machine and hit copy, the exact same worksheet is going to become spitting out of the machine. So no changes whatsoever. You want everything to be the exact same as the original. So our DNA is going to replicate in the nucleus for our eukaryotic organisms because DNA always stays in the DNA always stays in the nucleus in our eukaryotic organisms. So DNA replication will occur during interphase of our cell cycle, which is the preparation phase before mitosis and the actual division occurs. So if you need help with that, check my cell cycle video out. These two go hand in hand. Within our DNA replication, we're going to see a lot of enzymes. Remember, our enzymes are functional molecules, normally ending in ACE. They act as a catalyst, so they cause a reaction to occur. So these are going to be our 4 main enzymes in our story today. We have helicase, DNA polymerase, primase, and ligase. So helicase is in charge of unzipping the DNA. So it says unzipping genes, small segments of DNA. So the helicase is going to come through and break those weak hydrogen bonds. DNA polymerase, poly meaning many, polymer, many units. So our DNA polymerase is going to be the enzyme that builds the polymer of DNA. Primase is a starting point. So here I can see my primase having that starting point saying, all right, DNA polymerase, this is where you're going to begin replicating the DNA. So we say it's a primer for the DNA polymerase. And then ligase will follow that up. That's going to link the DNA molecules or the RNA molecule together, making sure it has that strong phosphodiester backbone and making sure it sticks together. So here's just kind of a run through of it. Our helicase unzips that double helix of DNA, breaking those weak hydrogen bonds. My primase comes in as a point of origin for the DNA polymerase to build. So with this, it's important to note that DNA polymerase can only work in the 5' to 3' direction. So on the leading strand, you can see this DNA polymerase is happy, just going along, adding the needed nitrogen bases. This DNA polymerase has to work backwards. So the primer will leave a mark. That DNA polymerase will run its course. And then it's going to have to detach and then go backwards to find a new location and work again. So the lagging strand, think of it as it's physically lagging because he has to do more work. He has to attach, detach to fill these fragments up. So leading strand 5 to 3 prime, lagging strand, he's still going 5 3, but he's going to have to be working backwards. So as we can see in this image, DNA polymerase working backwards, and we can see these little fragments here. So those are going to be called Owazaki fragments, which are named after the Japanese biologist who named the Okazaki fragment. So he named it after himself. So at the end, our ligase will come zip up those backbones, and we call our daughter strands semi-conservative because what you're going to see is they are half the original parent strand and half the new strand. So half of those new nitrogen bases or new nucleotides attached by the DNA polymerase. But they should be identical, which they are, having the exact same genetic information. So that is it for our DNA replication.